All right, welcome everybody to the seminar, the CARE seminar series, and today's seminar speaker. And welcome to all of you online. We like to know where you're tuning in from, so if you are amenable to it, please put where you're coming from in the chat. If you have questions online, audience, put them in the Q&A and we'll read them if there's time. We'll get through as many of them as we can at the end. Mulata and I are very pleased to welcome today's speaker, Dr. Theodora Pinou, who I met last fall when she invited me to do a talk at a road salt symposium, and who Mulata has known since 2004, 2005, because she was a student at Western Connecticut State University, where Dr. Pinou is from. Dr. Pinou is a professor of biology at Western Connecticut State University and a curatorial affiliate in vertebrate zoology at the Peabody Museum of Natural History at Yale. She received her BA, MA, and PhD at New York University in biology with a focus in vertebrate evolution and ecology. She's interested in the evolution and conservation of amphibians and reptiles and maintains an active sea turtle conservation program that studies the relationship between sea turtles and the vertebrates that ride them as they swim from nesting to feeding grounds. She's a research partner at the Leatherback Trust, works closely with scientists at the Peabody Museum of Natural History at Yale University, and her research has led her to be part of a team that has described new species and discovered new relationships between these new species and sea turtles. And her students have interned at museums, field stations, and worked closely with the Connecticut Department of Environmental Protection. There's another E in there. Energy. One of her students is working here at CARE in the analytical lab. Her talk today is titled Discovering Biodiversity, Going Beyond the Limits of Our Eyes. So welcome, Dr. Pinou.
Okay, so I'm just going to check. I guess you can hear me. Everyone can hear me. That's great. Thank you very much for inviting me here. And you know, we don't realize how close WestCon is to the CARE Institute, and so I hope that my being here is an opportunity for me to not only meet some of you, but to also expand potential partnerships and relationships. And I hope that this talk today stimulates some of those conversations. So as you heard, I am a herpetologist, and today I'm going to talk a little bit about how people like me in small institutions can actually bring or integrate their research interests with their teaching responsibilities, which has been a challenge, I think, for faculty in smaller institutions. Yet the situation now allows it for us to also be able to come out in the limelight, this whole interest in motivating and engaging diverse learners. And so today I want to talk to you a little bit about how I've done that. And my title is about biodiversity beyond the limits of our eyes, because I want to remind us all that some of the greatest discoveries remain to be found, and we need to stop thinking about just the big things that we see that are easy to see and measure and talk about, and start thinking about the little things that remain to be found that are just sitting there and have tremendous consequences on the balance of our ecosystem. Okay, so again, thank you. All right, so my outline today, I'm going to introduce you to sea turtle epibionts, because you may not know what they are. I'm going to introduce you to something called case-based learning and CURE, which is a little bit about how the pedagogical component of my talk. I'm going to introduce you to plasticity and how sea turtle epibionts are lending themselves to studies of plasticity. I'm going to talk to you then about environmental impact on marine systems. And my work which is currently going on right now on settlement, selectivity, and then of course, new discoveries. So this all begins in 2001 when I first went to Yale, and I was told, well, you've got to include undergraduates in your research. And for those of us who are herpetologists with a focus on snakes, including students in snake work is tough, mostly because you don't really know when you're going to find them or how you're going to find them. And so turtles are an opportunity because of their high fidelity. And therefore, focusing on sea turtles, sea turtle nesting, sea turtle conservation was a great way to bring undergraduates into the research arena. So one of the focus of my work at that time was to get out there, not just look at the sea turtles, but also explore what else we could find on the sea turtles. And this brought us to the concept of the sea turtle epibionts. And so here I am, everyone knows what a sea turtle looks like, but what you may not necessarily see often are the little invertebrates that are hiding in the cracks of the scutes or the sheds of the scutes. And this was our focus, like what's going on? What are we missing? What is there that we haven't noticed before? The other aspect of this was looking at sea turtles that are debilitated and the crusting on the sea turtles, and asking what is matting itself on these sea turtles? How is this happening? Why is it happening? And what is it doing? In other words, are the epibionts creating the problem, or are the epibionts a manifestation of the death of the animal, right? Like how is this system working? Okay, so we love sea turtles. We're going to start with that premise. That's why we can get out there. We can motivate students to go out. It's a public interest. It's of student interest. It's of interest because it's a big vertebrate, visible, high fidelity. So epibionts, or these organisms that are on, right, the animal, on the sea turtle, right, and you'll see, there we are, we've got some algae as well, are opportunistic organisms found, right, free living in the surrounding environment. And these are the words to focus on, is this term of obligates. Some are obligate commensals of specific host species. So we're going to talk about, or I'm going to show you examples of a lot of high diversity of epibionts. And then at some point, we want to focus on the obligates, because the obligates, I think, are what are really telling the story of what's going on here, or can be used as signals of what's going on. Okay. So when we talk about sea turtle epibionts, and this is the paper that I had offered when I was invited to speak. This is a paper by Robinson and Fowler summarizing the literature of the sea turtle epibionts, and they looked at all those sea turtle species and asked what was the diversity like across the different groups. And one of the things, and how many studies were there, right? Because, effort, of course, is aligned with diversity, right? So if you look more, are you going to find more? What does the literature tell us, but also is the amount of effort of the literature equal? So one of the things that we recognize is that not enough is known about Kemp's, Ridleys, and flatbacks. There just aren't as many articles. But more so, what's important to see is that high diversity occurs on loggerheads and then on hawksbills, and then we start seeing some drops in the diversity. So what is driving this? The other term that I want you to focus on, because many of you may not know what a lot of, maybe you remember from intro bio, the diversity of, biodiversity of all the living organisms. So we have a lot of the groups here. But the barnacles themselves are right in here, the arthropods, the theocostraceans. Okay, so we want to really just focus on that as well. So it's not just barnacles, which are often thought of when we think about epibionts, but barnacles are playing a big role. Okay, So what else, right? What else? Well, we found that we can have, leeches, right? Our traditional barnacles. Then we have other kinds of, arthropods, as well as some parasitic epibionts. And I'm going to come back to those because typically we've thought of the leeches as being contributors to herpes simplex on these sea turtles, right? We see sea turtles coming out with these large masses, right? But recently we're also realizing that there's other epibionts that are also contributing to parasitic disease on sea turtles. Okay. And then we also had experiences of life cycles. So are the larvae free swimming, or are they dependent on the surface? So are they living their entire life cycle on the sea turtle, or are they actually, do they have free swimming larvae so that as the turtle moves around, you're going to get an increase in the diverse settlement on a sea turtle, right? So in some cases, like on these gooseneck barnacles, we were finding that you'd have the adults, but you'd also have larvae with these very high numbers. So that's sort of signaling that something is happening on an individual animal. Okay, so the result of thinking about the biodiversity of sea turtle epibionts forces us to also realize that conservation of sea turtles is not just about eggs, right? It's not just about nests. It's not just about the beaches. It's also about thinking about whatever diseases or parasites or other interactions are occurring in the oceans. Okay, so this concept of what else is going on that we need to be thinking about as the climate is changing, as the presence or absence of a particular diverse community in the marine system. All right, so another important finding is that the diversity of epibionts, for whatever reason, happens to be higher on sea turtles studied in the Atlantic versus sea turtles that are studied in the Pacific. So the richness, the biodiversity richness is higher in the Atlantic versus the Pacific. Okay, but what's key really are the obligates. The obligates, the barnacles, are the same on both oceans. The difference is this non-obligated group. So for whatever reason, there's just more going on. And we're not going to get into the differences between the Atlantic and the Pacific Ocean, but for many reasons, the Atlantic just happens to have, this richer invertebrate planktonic mixture than we see in the Pacific. It has to do with the depths and the temperatures and the age, and there's a lot of factors. But it's playing out also on the backs of the sea turtles. Okay, so we have diversity, we have species diversity, we have a system, we have a host, we have these interactions between obligates, non-obligates, and then, one says, how can we take, we want to know why, we want to know what's going on, we want to try to understand the system. It's more work that's going to be done in 1, 2, 3, 5 years. It requires repeated sampling, it requires taxonomists, it requires modelers, it requires a lot of work from a broad community. So how do we speed it up, right? And one of the things that I want to introduce you now to is how do we take some of these interesting questions that we have about the relationship between sea turtle epibionts and the diversity and their relationship to sea turtles and sea turtle conservation and integrate it into our classrooms. How can we move the research forward faster, plus enrich our classrooms. And so I want to introduce you to something called case-based learning and cures. And these are well-known pedagogical tools, right? Case-based learning. What happens in case-based learning? Students are going to apply knowledge. Teams work together to find solutions regarding disciplinary problems. It's more student-centered. Basically, the faculty member takes a back step, right? And the students in teams investigate the case, the problem, look for solutions. And of course, there's going to be disbelievers in here, like this doesn't work, they have no knowledge, how are they going to do this? But our students are pretty crafty today in finding information. It's not that I do nothing, you guide the conversation, but the students are coming up with the right kind of information to help guide the solution, okay? And I just want to, point out to you that this is a legitimate teaching tool, and it's one that's very closely aligned with project-based learning, which maybe some of you have heard more about. And project-based learning is also a strong tool that's, highly recommended in secondary education. It's one where we're trying to get the teachers to do less teaching and students doing more work, right, with a guided approach. But it requires some fundamental sets, right? It requires that the work that they're doing, the problem, right, the case is authentic. Well, the sea turtle work is authentic, okay? It requires that there's a question, inquiry, that the student can ask the question. And given the volume of work that we were using, there were lots of questions in there, whether you wanted to talk, ask taxonomic questions, relationships, modeling questions. So the students had choice of voice. The students are driving a lot of the work that's happening. Collaboration is key, right? Community partners are key. So museum was included, right? Different scientists collecting in the Atlantic, scientists collecting in the Pacific, scientists at different field stations, right? A wealth of literature of people with the sea turtle programs and permits. So the students are being introduced to these collaborators, right? To these community partners. They're engaging with them in an authentic way. And this is resulting in employable skills. And I think this is really key. And right now, what's driving a lot of the DEI conversations, right? Diversity, inclusion, equity, and how are we going to motivate, engage diverse students in STEM, in interesting questions. And the philosophy is it doesn't just come from the faculty member or the teacher. It's got to come intrinsically from the students. So what I can provide is motivation. I can provide a really good story. I can provide a good case. But the student is going to help me come up with the solutions in a guided way. And I know that sounds like a lot of fluff, but it really does work. And my job today is to show you how. Not only am I telling you this, but some of you who are in the grant writing world probably have heard of cures. Right? The CURE, right? Course-based undergraduate research experience. That's exactly what's going on here. And these are well-funded. So this project-based learning, this case-based learning, this is a tried and true pedagogical tool that's shown to increase engagement, motivation, commitment, build skills, and build knowledge that will help professors, researchers move their knowledge forward. Okay, so what are the cases, right? Epibionts are opportunistic. I'm just going to throw out a couple of ideas, right? They're opportunistic, right? They're not necessarily obligate, right? They're just landing on a surface. And, here we go. We have little Podocerus. We have them in the mats of the algae. right? Is Podocerus there because the algae is there, right? Like what is going on? What's the dynamic? We also have them nested in the cracks of the barnacles, right? And so this is going beyond your eyes because you see the barnacle, you see the sea turtle. What you probably don't see is that there's more, right? So what are they doing there? Is this just an opportunistic situation, right? What's the story about the obligate attachment? Why do they attach in an obligated way? And why are they attached specifically, right? Certain barnacles attached to certain species of sea turtles. What's going on there, right? So here we have our gooseneck, right? And you see all these little sand granules. And here we're trying to peel off these barnacles that are really tightly attached to the back of the turtle, of course, which adds itself to how does that happen, right? What's the cement that's being used that's really creating this tight attachment, which is another case, right? Like, how does this happen? What's going on? What's the biochemistry, right? The biophysics that's involved here. Okay, so I want to bring you into this case-based study or this cure. How did it get started and how was it used and the outcomes that are, right, the metrics of all of these things. So, right, my grad student, Christine Lerner, interested in sea turtles, decides she's going to look, and she also already had a relationship with Mote Marine Labs. So the question was, could we sample from Casey Key regularly, several occasions, and do the, you know, the analysis of the epibionts, the biodiversity, what's there, right? We didn't even know what was there. So we had collections of material, different turtles, different years, much more than any one single graduate student can handle. So what do, right? We bring in our students, right? This is a research class, right? We have these at WestCon where all students have to do a research project. They can either do it as an honors thesis, or we actually have a course on design and research, right? Where the faculty member kind of drives the research agenda. And this was this was the platform for this cure. So, you know, students are learning how to handle the material in a curated way, right? Because you don't want them to mix them up. You want them to sort it. You want to make sure you maintain the integrity of the sample. Who's the host? What's the year? What's the diversity? Right? And so all of this is going on in the classroom, right? 101 turtles in a semester. Now, I point this out to you because one grad student, this is a lot of work. It can be done, but it's a lot of work. But now you're integrating the undergrads in an organized way, in a facilitated way. It's benefiting the grad student because they're learning something about pedagogy. It's benefiting the students because they're involved in authentic research. Okay, and of course, you know, they came up with they're going to learn they're going to learn some statistics. They're going to learn how to calculate biodiversity. They're going to learn to talk about it. They're going to learn about the, how to identify some of these invertebrates. So they're learning a lot about epibionts and how to explain what they have in these connections, right? And so we get a lot more information and a lot more questions, very few answers. And this is, this could be the frustrating part of a cure because you've got a lot of minds, a lot of ideas, and let's remind ourselves of mythology, right? Out of chaos comes beauty, right? So you need to have the chaos, you need to have the confusion, you need to have it all there. And then you need to start teasing it through and thinking about things. And these are just different pictures to highlight the diversity of what is in this Atlantic collection, this high diversity. And now I want to introduce you to Molly. So we've had the cure. We've had all of this work that's been done, all of this information that's been collected. And now we take a highly motivated thesis honors student who says, I want to make sense out of it. And she focuses on it. And she asks the question. She also was able to get another sampling from Moat Marine Lab. So she was able to look at epibionts that were collected off of turtles from the same beach after the oil spill and before the oil spill. And really asking the question of, do we see a localized change in the, in the planktonic layer, right? Where these turtles are swimming, and what was the possible community that could have attached itself on these sea turtles, and was it different? And so when we look at the PC, the principal component analysis, we see that, okay, we're getting some kind of signaling here between, the darker points, which are the pre-oil spill, and then I don't know if the reds are coming up as clear, but the majority here are red, which are the post-oil spill. So there's some sort of signaling that's going on. And one of the things that she was able to work with the faculty closely on is teasing out the fact that, in fact, there was a difference in the obligates. So when she looked at the Chelonibia, which is one of the obligate barnacles, and I'll show this to you later in future slides, she was able to show that once after the oil spill, one of the barnacles that you don't see a lot of, all of a sudden increased in frequency on the turtles, and the one that was common disappeared. Okay, and when that one disappeared, you also had a higher diversity, right? So here it is. So Testudinaria is what we would expect to see, but in the post, we started seeing Coretta and a whole bunch of other things. All right, and so this is, sort of the schematic, and I don't know how well the colors are showing up, but here are all of the Chelonibias. Testudinaria is the most common, and when Testudinaria seems to go down, Coretta goes up along with everything else. Okay, and we were able to get this published. So my point is, what started out as a cure, what allowed us to go through 100 and some odd turtles, all this information, educate all these students on some statistical analysis, talking about biodiversity, informing them about what's going on in our oceans, ultimately ended up as a paper as well in an interesting concept about how the environment can influence, right? Now, we didn't test it. We didn't test the oil on the epibionts, but we did see that there was this association between, the pre and the post, and we will continue to monitor that. So cure learning again, right? These undergraduate research experience, course-based undergraduate research experiences, right? They're authentic, add new knowledge of the PI, right? So builds the research agenda of the PI, and you're able to scale it to meet diverse learning venues, right? And this is how we went from the class to then the Molly scenario, which allowed us to ultimately publish. So these, project-based learning cure practices are aligned with best practices for STEM engagement. And so, to just close this piece of my presentation, it's important to realize, in a place such as Cary, where there's a lot of research that's going on, a lot of data that's being generated, there are opportunities to partner with, faculty who may be aligned with your work, that have the students that can use your data, not only to educate, but to also move through the data faster and get you to maybe a conclusion faster, right? And you're also contributing to the betterment and the STEM engagement and all these other things that we're supposed to be doing besides just our own work, right? Okay, so what about plasticity? I mean, what has some of the sea turtle epibiont work that I've done, how does it lend itself to this plasticity question? And I want to introduce you, right? Phenotypic plasticity is a change in the organism's behavior, morphology, physiology in response to some sort of unique environment. I want to focus on morphology, and I want to turn your attention. This is a CAT scan barnacle. This barnacle was embedded into the shell of the turtle, and we actually were able to get it, capture it. And it's important because, you know, this is Stomatolepis elegans, and I'm going to tell you the story about Stomatolepis elegans. It's a type of barnacle. It is an obligate barnacle, and there are two Stomatolepis lepi that we were interested in. One was elegans and pregustator, and honestly, they have lots of morphological differences. People could say that they could tell them apart. We did not feel that the morphological descriptions were holding true because there was so much variation. And the one thing that seemed to be, and this is just more variation, right? One thing that seemed to hold true was, well, pregustator was always isolated from the gullet of the sea turtles, whereas elegans was always isolated from the carapace, right, of the sea turtle. And could that really be a gene flow barrier? I mean, could you really have species level distinction based on, I mean, they're all in the same marine system. How is it that one particular species is attaching to one place and another species attaching to another place? Like, you know, that was a hard thing to be able to think about. And so just to help you understand how you're seeing these things, right? Here they are attached to the carapace, and then we have, right, the stomatolepi that are attached, right, in the, and let me tell you, it's not easy to get these, but you do, right? So here we are, right? Okay, not easy, right? You pull straws on who's going to be responsible for this. But is this possible? I mean, it's possible. Does this address the morphological diversity that we see across these two species groups? So, you know, at that time, what we did was, okay, you know what, let's just do the genetic analysis and find out. And of course, you know, we limited, this was just COL1, 600. I mean, some of you must be looking at this thinking, oh my God, this is archaic. But what's important, and I'm just going to move through this because this is the picture I want to show you. When you look at elegans, right? So here are all our turtles, and here is Stomatolepis, right? Pregustator, elegans. What's going on? Well, elegans and pregustator seem to be nested. And as we expanded this and looked at this more, and of course, you know, you're probably also looking at these distinctions here, which have since been removed from Stomatolepis and have been given their own new genus names, right? But this is the key thing. And the key thing that we found was elegans pregustator is the same thing. They're the same species. They're just manifesting themselves morphologically different when they're attached to the shell or when they're attached to the gullet. Okay, it doesn't make a difference. So what's controlling this plasticity? What's controlling the morphological change, right? Depending on whether it's attached to the shell or it's attached to soft tissue. Like, what is going on, right? And we don't know. All right, the reality of that is we don't know. We do know that Stomatolepis elegans is highly diverse in its appearance, and it responds to the environment that it's attached to. So what is going on? Environment is clearly playing some sort of a role in whatever's going on with these epibionts, because we also saw that given some chemical change in the environment, we get a drop in one species and an increase in another. And between the two Chelonia, and this is going back to Molly's project, these are two different species of Chelonia. We know they're good, distinct species, right? So what we found with Molly is we're losing the testudineria when we're increasing the Caretta. What's going on with the Stomatolepis and the morphological change? We really don't know, and we're still testing a couple of scenarios, but it's probably the substrate, right? Because the way the barnacle attaches and then extends its attachment into the substrate, there's obviously some sort of control, right? There's some sort of interplay between the two. Okay. So again, this is just to remind you of the pre and post oil spill work and that response between the shift in the community. As one obligate decreases, we see colonization of a whole different group. So what is this telling us about future changes in our marine environment as, for example, climate change happens, right? What is going to happen to the invertebrate diversity over time? Okay, so a little bit about settlement selectivity, which is another project that's going on right now. You know, we talked about this plasticity between individuals of Stomatolepis as they land on the landscape of the turtle. And so we asked ourselves, what is it about the landscape of the turtle? And so we did some microdermatoglyphics of scutes of lots of turtles, right? Major groups of turtles, all the sea turtles, and at different magnifications to sort of see what is going on with the actual scute. And, you know, these are cells that are composing, right, the scute under SEM. And you'll see that we can see the cell outlines, right? Sometimes they're juxtaposed, sometimes they're overlapping. And as we go up in magnification, we start to see that there are pores. And this is not an unusual thing for reptiles. I mean, you know, we have this in snakes as well. I had done a lot of this work as a grad student on snakes. So we know there's a sense that that the animal gives off certain compounds through these pores to allow them to become more hydrophobic, to be able to, you know, maintain balance. Who knows, could also be an immune responses are going on here. The point is that there is an opportunity for a barnacle to land and attach itself in some way, shape, or form. The problem is that when we try to align the characters to a known phylogeny of relationships, right? So here are the two Lepidophytes species, you know, and you know, this is one well accepted clade, and then here's the second one. It doesn't explain the epibiont susceptibility. So in other words, Halonia mitis has a high diversity of epibionts, and from what we know from nadir does not have a high. This is the Australian flatback turtle. And you know, we wanted to believe that pore reduction was what was driving, right? So as the pores shrink, the amount of attachment is going to decrease. But because that's also true for the Kempi. Kempi is least susceptible. The problem is that the hawksbill turtle, which has overlapping cells and pore reduction, has actually quite high epibiont attachment. So we're still working on this, but this is one way that we're trying to understand what's going on. What is interesting is that if you look at terrapins, they also have the same kind of pore structure, cell pore structure. And when you look at more of land turtles, and this is the pond turtle here, so you can't say it's an aquatic terrestrial thing. These don't have pores. So there's something going on with this this paradigm, but does it explain our epibiont diversity? Not yet. We're still working on it. Okay, so I want to talk a little bit about the last part of my presentation, which is on this topic of discovery, because this was a lot of fun. And this was a reminder that we need to go back regularly to our museum specimens. We need to look closer. We need to be thinking about what are we missing, right? What's, how do we know everything? Is there more to learn on things that we've already looked at, right? And so here we are working in Costa Rica, right? And everybody's on the field station thing, and you're there, and it's easier to work there and not have to worry about permits for certain exports and yada yada. I can tell you about all the benefits of working on site, right? But what's interesting is we realized that every sea turtle had its own unique diatom community. And you wouldn't have seen this if you hadn't really looked at, when you wash the scutes out, or you try to coat the scutes and then look at them under the scope, and you realize, oh my God, wow, okay. So we discovered new species, and of course, you've got to work with somebody who knows these diatoms, but it was quite fascinating to see that there was unique communities of diatoms on sea turtles that were sharing oceans, right? So some kind of selectivity is going on. And what was really unique was also finding that you could find them on museum specimens. So, we went back and we could do it. So there's a lot of material that's out there in museums, and it's a question of looking, okay? All right, so in closing, right, discovering biodiversity, going beyond the limits of our eyes, is just to remind everybody that there's, again, a lot to be found. Look beyond your eyes. Look beyond your eyes in terms of things that exist, things that you've sort of shelved and collected and maybe walked away, like what's there? It's a record of the past, right? If you revisit it with a new set of eyes or a new set of tools, might you see some new information? And thinking about the genetic level diversity and what that could add to biodiversity. And I think a lot of you already recognize that, right? But just really taking a good look at species level, population level, genomic diversity. And how it plays to the morphological, right? And thinking about plasticity and thinking about how things are diverse, and yet when you look at their genomics, that they're actually the same. It's just that they're responding differently to their environment, and how that plays into biodiversity in a changing planet, right? And of course, beyond your eyes in the sense of the microscopic. And I want to close by saying how important this kind of work is to engaging and motivating diverse learners. I remember so many of the most influential scientists for me were people who said to me that what got them involved is Treasure Island, right? What got them involved in what they love to do is Treasure Island, especially the person I work with at the Peabody. That's his story. Treasure Island is what did it for him, right? That's when he decided he was going to work in the Caribbean. Great stories that naturalists wrote, right? When I teach my field class, I use a naturalist in Nicaragua, and I've gone to Nicaragua with students, and you really follow in these trails of the writers, and you talk about the differences. And Some great natural history works here, even in the Northeast, right? Every time I drive up, I think about Bard and Gerard, right? And all the original work that they did in this area. Teaching from those perspectives, coming out of just lecturing, but really trying to find new ways or go back to the old ways, right? For a new generation of students to excite them. Being in the field, really good books with lots of descriptive work. I mean, if you read field notes from the old, the old timers, they're beautiful. They're just beautiful works. And there's information there, but it's not so rigid, right? But it's very exciting and very motivating. So I want to remind everyone that none of this happens without a group of a team, right? It's all about teamwork and all the people that are involved, both local and internationally, and the importance of building strong collaborations. And of course, nothing can be done without funding, right? That allows you to keep those smiles. And thank you, right? This is the cloaca of sea turtles. So, you never know where you're going to find diversity, but it's there. So thank you very much. I'll take some questions.
Thank you for a nice seminar. There's plenty of time for questions. Thank you, Matt. And I want to remind the folks in-house that we're going to have to repeat your questions because the online audience can't hear you. So if you could please keep your questions short, maybe shorten the preamble that we often do before a question. But so yeah, open for questions. Josh, you had your hand up.
Sea turtles can live a very long time and have indeterminate growth. So have a huge range of size and control for size and the surrogate for age on the density diversity of epibions.
So you're asking me if
Repeat the question.
well, yeah. So the question is if we've considered size and age in our work with the epibiont diversity. And the answer is yes, in an indirect way, because a lot of, well, the work started on nesting females, so they are all of one age group. And then we, we also have studies that we've done on leatherback turtles because they're so high in their fidelity in the Pacific. We actually cleaned off individuals, cleaned them off, and came back, not because they don't come back the following year, but they'll come back the year after that. And we didn't expect what we found. They were completely covered again. So I think what we're capturing, obviously it was an opportunistic landscape, and the epibionts were able to attach again. That was our understanding. So I think when we see the animals and we're cleaning them off and we're getting this diversity, we are clearly capturing a point in time, right? And we don't understand everything about what that point in time is. But we do know that if the carapace is clear, there's going to be attachment. And there's going to be a limited amount of attachment. Because if you go over it, the animal can't swim. It loses its buoyancy, it loses its flexibility. In fact, there's some great videos of loggerheads actually banging up against reefs to knock them off. It's quite fascinating. So to control, I don't know if that answered your question, but the answer is yes. And the juveniles don't seem to have an impact of epibionts, and probably because they're shedding their scutes on a regular basis and these are coming off. So that's about what I can tell you. We have in another study now that I'm thinking about it, we also did include in Costa Rica, we actually did in water work. So we were able to look at males and females. We did not find any differences. So we feel pretty good about the age and size and the story we're telling. Yes.
Go ahead.
I was wondering if you looked at microbial epibionts and whether or not they are facilitating the attachment.
Yeah, so you're probably, so the question is, have we looked at microbial layers and the mats, right? To see if they're, and the answer is not yet. It's on our to-do list. Right now, we're looking at some of the algae that's on there. It's really hard. It's really hard because you can't raise these. It's very hard to raise the epibionts. We've just started learning how to do the barnacles in the lab. If you can't control some of the growing, it's hard to try to, it's hard to do some of the actual testing.
I'm just curious if your differences in your species might be because of the microbiome ability to dominate.
Go ahead.
I also had a question about species differences. I know nothing about the habits, but I'm curious how they spend their time among the different species, and if them spending time in certain latitudes or habitats or depths might explain some of their variation.
Yeah, so you're asking about, the question is about behavior, migration behavior, and whether migration behavior can explain some of the diversity that we're seeing. And that was exactly what Christine Leonard was focusing on. And we have, one of our studies we had tagged the sea turtles and then looked at their epibionts. And we did not find, and that was kind of a shock. Like we really thought that the sea turtles would be carrying representations of where they've been feeding, right? And either we need a bigger sample size, or we're missing it. Certain things are there, like, you know, you'll see certain signals of deep, you know, of depth, but we're not ready yet to, I think, look at it that way.
I have a couple of questions. The unique diatom communities on the turtles, that's absolutely fascinating. Any idea why each turtle has a different community?
No, and that was, so the question is about the different communities of the diatoms on the sea turtles. And I mean, you know, I'm not going to stand here and pretend that I'm, you know, like that's not what I do. But there's a big interest in these pores and trying to find out what's going on, right? What is being given off. But that's not an easy thing to do, you know. But that's what we're trying to hone in on to see if that's partially what the explanation is. But it's not, you know, I was going to say age, because like we looked at some of these museum specimens and the diversity held true, right? But then again, you know, how old are these museum specimens? I mean, 50 years old, some were 100, right? But now still, you're talking about water, you know, climate or a local environment that's 100 years. I mean, I don't know. Is that a scale that's? So no, we don't know.
Okay. I want to remind the audience online that you could put your questions in the Q&A. I don't see any questions. I hope that's real and that I'm not just misreading this. Are there any other questions? Peter, go ahead.
So you have the Epibias from 106 turtles, gave to students. And did and so I'd like to hear a little more about what you did with that. Like, so did the students then do comparative studies? Did they have individual projects?
So great question, right? Because that was my panic. I was like in a panic, right?
Repeat the question.
Oh, yes. So the question is, you know, what is actually happening in the classroom when the students have access to 106 turtles that represent authentic data, right? Authentic time. And, you know, yeah. So as the PI, you panic. right? Because you're like, what am I going to do? I have to let go of this? No, right? But you got, yes, you make that commitment. I had tremendous, I have wonderful collaborators at the Peabody Museum, and they were able to come in. So the ratio of students to faculty, if you want to call it, was very low. Each student owned a turtle, basically. And so the student had the container with all the material. The students all had keys. And I reminded myself when I was at the museum, used to give my son, because it's what people do, like museum people do, like you just give them some bones and they sort them out. Like kids are really good. Young people who don't know, like people who don't know much, like volunteers, museum volunteers, they can sort. People can sort, right? And they have their own mindset on how they do it. You can't control it, but believe it or not, they're really good at finding characters and putting things together. And they, even then they don't know a copepod from an amphipod, they learn pretty quick, and you just have to manage its quality control. So they were, once they can handle the specimens and they separate them out and they know what they are, then there was a collective data table, a class data table. And when you do this, it's a biodiversity table. So you had the turtles and you had the diversity and the numbers, the frequency. And that's when it gets interesting. That's when you say to them, okay, what questions do you want to ask? I mean, this is now, you're observing this. What questions do you want to ask? And so some of the questions that they asked was seasonality, turtles that were, sampled in June, did they have the same diversity as turtles that were sampled in August, for example, right? They asked questions about years. Turtles that were sampled in 2000 and blah, were they the same? Did the biodiversity, was it any different than what was sampled the following year? And in all of those cases, there was no significant differences. It was only until Molly added the oil spill, and she had an extra year of material, because what had happened on that year, we didn't get the full collection, but after the cases were all done and settled down, we were able to access all of that material was sent to us. So she actually had a full year where she could go back and look at that. And that's when the signal became somewhat apparent, right? And we have more. And what I'm hoping to do is see if over time, these new samples are more like the pre or the post or something else, right? But to answer, I don't know if that answers your question. I mean, I'm going to be very honest with you. It was not an easy thing for me to do. Not easy at all, especially, and this is a true story. We came into class and there was a turtle missing. Nobody knew where the container was. And I was not happy. And I don't think the students ever have seen me like that, or the grad students or the faculty, but I was devastated. I mean, one sample is a huge thing when, you're doing this kind of work. And it was a teachable moment because one of the students came up and admitted that they had been in on the weekend, they had worked on it, and this is where it was. And I really opened up and I explained to this is authentic work, right? This is the authentic, this is the teamwork. This is when you've noticed that I'm disturbed by this, and someone's honestly, you're not afraid to stand up and say, I came in on the weekend and I did it, right? It's not a bad thing, it's actually a good thing. And I think the students saw the relief, like it was a teachable moment for everyone on how important it is to be transparent and honest and collaborative. And honestly, they hired this young woman at the museum afterwards as a curatorial assistant. I mean, she was one of the, that honesty. I don't think young people, undergraduates, necessarily realize just how important the honesty and transparency is in authentic work. It's not about, can you do R? It's not, you can learn that, right? It's not, but it's these other skills that are so incredibly important in job training, in teamwork, in research. You know, the honesty, ethics, all of these things.
Well, we've hit the top of the hour, so I'm going to bring the questions to a close. Dora will be joining us in the lunchroom for lunch, and everybody is welcome to join and carry on this conversation. And let's thank her for a nice seminar.